Well, basically what I study is the gut-brain axis, and that's a difficult word already, but there is a connection between the gut, your intestines, and your brain in relation to neurodegenerative diseases, which includes Alzheimer, dementia, but also Parkinson's disease, those patients who suffer from motor problems. And especially with the last disease, we know that these people suffer from gut problems far before they get diagnosed. And people are getting aware now much more that in dementia, there's also associated with gut problems. Well, basically, the field started off with the Parkinson's disease because it was already recognized when it was described that there was a gut problem. And then we got the hype of the microbes in our intestines. So I think doctors working with Parkinson's disease patients were more aware of maybe we should look at that. And we know there is a difference in the composition of the bacteria in the intestinal tract in Parkinson's disease patients compared to healthy controls. And that's been tested now all over the world. There's not one bad bug popping up, unfortunately, but there are huge differences. And we see it's more pro-inflammatory, so there's an inflammatory process going on. Your gut is a bit leaky. So people think that bacteria can travel and reach the circulation and then get maybe towards the brain and induce inflammation there. And we know in the brain there's inflammation and there is misfolding of proteins in neurons, and then the neurons die. So, and that causes the motor problems. And a similar process is going on, slightly differently, but also in Alzheimer's disease. You find a similar pathology, and so people are really picking it up, and we have preclinical work investigating really the role. You can deplete the microbes by antibiotics and see how the disease develops, but you can also treat, for example, with probiotics or fibers, which beneficial bacteria like in diets, and you can find effects on the development in the disease models. So the next step is to do this in man, obviously.
So what can be, for you, according to you, the future perspective for medical food in this kind of disease?
Well, basically, I just got this question. Fecal transplantation is sort of quite hot, but I would be very careful with that because we basically don't know what is a healthy microbiome. So food interventions with probiotic strains, but we should really investigate the difference between the strains because one strain is not doing exactly the same as the other strain. And we should also try the fibers, which make the beneficial bacteria grow, but we should take maybe a step back and first investigate what's out of order in relation to your bacteria, and then design a personalized treatment. I think that would be the best future.
Can you elaborate a little on this brand new, exciting new or not, that we have microbes in our brain?
Yeah, I've been struggling with that because I was thinking, why are there microbes in my brain and why like the microbes be there? But to keep it really simple, we have the brains with the neurons and we have the cells around it to keep the neurons in place, but we also have like open spaces in the brain, and in my opinion they are very similar to our gut. So you have the epithelial cell lining there, and basically they store all the rubbish which is collected in the brain to get rid of it. And that compartment, I'm a believer, it's not been shown that maybe bacteria live there as well to tune the immune system, because the brain also has immune cells and they need to be trained and they need to be controlled. So maybe the microbes in our brain are there to control it. So yeah, if you have the good guys, you're healthy and happy. If you have bad guys, you can get neurodegeneration or you get mood problems or something like that. But that's very speculative. We still need to research it. I was here the first time as well, and I was so pleased that finally we have an event like that, combining people from the microbiology field, from the immunology field, from the neuroscience, from the clinic, dietitians, and people who are in nutritional sciences, because that's the way how we should go to dissolve diseases. So I really like it, and each last time I was flabbergasted, and this time again, because there's so many nice young people doing great research, the posters with all these new exciting things. So hopefully we'll have a third one in 2 years. So there was an enormous development. When I started working on the gut-brain axis, not in neurodegeneration, but neurodevelopment, people laughed at me because I did animal models for food allergic reactions in the intestines, and I was measuring behavior, and I noticed that these mice were showing autistic-like behavior, and people didn't believe it at all. And then it started to evolve, and then also with the developments of the techniques to measure the composition of your gut bacteria, it really speeded up. So, and also looking at disease in a more systemic way than only a problem of the brain or your intestines, that's also gaining grounds even in the clinic, and patients are more aware of it. And patients do a lot. What they can change in their daily life is exercise and diet. And they experiment. If they have a chronic disease, they experiment with it and they ask the doctors questions. So they need to be trained in a novel way. And you see that also developing, at least here in the Netherlands.
Yeah, sure. My presentation from today was actually about our findings in the Parkinson's disease context. So what are the changes in the gut microbiota in Parkinson's disease compared to healthy controls? And we were also having a look at the very prodromal stages, namely the RBD, so this is the rapid eye movement sleep behavior disorder diseases. And then we observed already some nice changes in the abundance of Akkermansia, for example, which is a mucus foraging bacteria. And this mucus foraging is actually increasing the permeability of the gut. And like this, you can have no toxic reactions already happening in the gut that might migrate then via the enteric system inside the central nervous system and then launch the pathophysiology of Parkinson's disease already at the very early stages. Yeah, so Akkermansia has been described of being beneficial. However, in our study, we have seen that it's linked to the mucous erosion, so actually it has not that of a beneficial impact of your health. So this is at the moment not yet clear. So what are the actual mechanisms behind? So whether Akkermansia now, what are the functions of Akkermansia now in this study or in Parkinson's disease in general? But so what we were interested in is actually looking beyond now the baseline studies that we have been doing. So what is actually the function? So we are actually analyzing metaproteomics, metabolomics in order to elucidate, so what could be the really impact behind that? Instead of just looking at the abundance levels, we are also looking at the functional level at this case. Yeah, so Akkermansia is actually known to be significantly higher when you have constipation. So constipation-based increase is observable, but in our study we did not observe, so we actually checked for confounding factors like constipation, if this impacts now our results. But we could not see that the differences we could see in the Akkermansia are actually due to constipation. So this is, we could exclude this at least in our study. The most important part of the work we are presenting is actually that already at prodromal stages of the disease, so mainly in the RBD, that are actually converters. So 80 to 90% of the RBD patients are actually converting to xenoclinopathies such as Parkinson's disease. And you can observe that these changes in the abundance, for example, of Prevotella and other bacteria, they're already present at these stages, like 20 or more years before the actual disease happens. Yeah. So I see an added value of this conference here. So it was the first time I'm actually visiting this conference. So I was quite happy to see all these kind of interesting talks, also in different neurological diseases such as Alzheimer's. There was depression that was also showing some results that we could also see on our side. This might lead to an added value on our research as well, because we are actually focusing maybe too much in our Parkinson's disease research, and so it might also be of a great value also to have a look at the general effect in the neurodegenerative diseases, to exclude that our changes are actually due to the overall general effects and not specifically to Parkinson's disease. So in terms of clinical applications, I would actually be in favor of having like a really early stage diagnostic marker. And this we have actually shown with our RBD data, the first indications that the disease already starts at very, very early stages, so decades before the disease actually starts. So this might be one of the potential targets for like a targeted biomarker for this disease stages that we can have then also in therapeutics way to actually treat the disease directly.
Yeah, I work a lot with the role of the gut microbiota for Alzheimer's disease, and I try to look on the molecular mechanisms for how bacteria might actually influence the brain and neuroinflammation. So I try to modulate the diet and optimize health effects of diet by looking at how the gut microbiota changes in response to the diet. And then we look at the brain and see if it prevents also neuroinflammation and Alzheimer's disease. And our main findings so far are we are looking a lot on lingonberries and how these different types of berries really, how they can influence the brain.
Lingonberries are kind of cranberries, right?
Yes, they are sort of red berries that are found in the Nordic countries, and they have very good effects on diet-induced obesity and atherosclerosis and improve the blood lipid profile. So we're looking now into if they can also impact the brain, and it seems like they can increase the number of synapses in the brain, in mice at least. And we also see an increased number of mitochondria in the brain. So it seems like lingonberries can change the microbiota in a way that actually boosts the brain. So, yeah.
And how can you extrapolate this to human?
Yeah, we're actually doing, we're planning now a dietary intervention in humans with different types of berries also. So we hope that we can see some of the same effects in humans. And then we are aiming for people who are not completely ill with Alzheimer's disease, but instead we try to go in and prevent the progression of disease. So we will look at people with mild cognitive decline and then give them different berries for a study period.
And why are these lingonberries specifically so effective? Is there a certain compound that's responsible for it?
Yeah, we don't really know. We try to explore a little bit if it's the soluble fiber fractions or insoluble fiber fractions in the berries that are important, but we haven't really been able to see what it is. But probably the fiber fraction is important because it changes the microbiota composition in the gut. But it also contains some benzoic acid.
Can it do with prebiotic properties, basically?
No, it's more linked. I would say it's like you can use it as a preservative even. It has even antibacterial effects.
Okay.
So it's an acid that seems to be also involved actually in major depressive disorder. I saw one of the posters here that reported that a metabolite of this benzoic acid is actually reduced in people with depression. So maybe it's involved also in this work. I see that we will move into more of the molecular mechanisms for how bacteria can actually affect the brain, and all the different signaling that must go on for the bacteria in the gut to affect the brain. And I think that we will also have more effective dietary regimes that can change the microbiota in a very targeted manner to affect the brain.
And do you see any pitfalls as well? You know, what types of things shouldn't you do with this research, or which is?
I mean it's a complex area, of course, and it's we heard today also about transit time and that can sort of be a confounding factor in microbiome studies, because if you have a constipated gut, you will have a different microbiome profile because of it. So I think that's an important thing that I've heard by many researchers bringing up now, that we need to be aware of that. And so it's all these confounding factors. Also the medicines that you eat affect the gut microbiota. So
how do you streamline these studies?
I mean it's very easy in animal studies to make it very standardized.
That's of course a confounding factor by itself.
Yes,
the gut microbiota of mouse is totally not visible.
So it's we need to do both human studies and animal studies, and try to get some sort of consensus of what's going on.
Any key messages that you take home from this conference?
Oh, quite many. I see that many people are looking at bacterial metabolites, short chain fatty acids and bile acids, and I think that will be very important to look at not just the bacterial profile, but what the bacteria are doing, and different metagenomic studies will also be very important.
We know that abnormal proteins present in food, in the case of mad cow disease in beef, can cause proteins in the body to adopt the wrong structure, and this can cause the disease, which is called mad cow disease in this case. It's been discovered that there are also proteins made by bacteria, and these are bacteria that live inside the body, and these proteins have a similar conformation to the conformation or structure of proteins in the brain and in neurons that are found in the gut. And we have demonstrated that these proteins made by bacteria in the body can cause the transformation or restructuring of the neuronal proteins so they adopt this disease-causing conformation. Also, these bacterial proteins found in the gut can cause inflammation in the brain, and both inflammation and abnormal protein structure are important features of Alzheimer, Parkinson's, as well as ALS or motor neuron disease. Before we can talk about a cure or a prevention, we should address the question of what's causing the disease. And 1% of Alzheimer cases are caused by a disease-causing mutation, which is usually of early onset before the age of 65. However, for about 99% of cases of Alzheimer's disease, we don't know what the cause is. And in these 99% of cases, the cause is not a gene. It's likely to be some environmental factor. And what that environmental factor is, we don't know.
Actually, you hypothesize that possibly neuroinflammation could be one of the primary reasons or causation of the disease?
Yes, certainly. There's very good evidence for that. And it is rather exciting to consider that there are a lot of things people can do to lower their inflammation in the body through diet and other means.
So what would be the next steps for your research based on what you've done so far?
We are continuing to do studies in animals, in mice, in animal models of these diseases, as well as in humans, to look at bacterial populations that are present in the body and to see how these abnormal proteins are being made and what we can do to stop them.
Based on what you've heard during the last 2 days at this Mood and Microbe conference, what triggered most of your interest? Is there anything that you felt could enrich your thinking for new research in your field?
Yes, I've met lots of people who are doing advanced and exciting work in this field, both from an immunological and bacteriological approach. And this kind of meeting is critical for this area because it really involves the merging of different disciplines. And there are a variety of them. There's microbiology and biochemistry and immunology and neurology and psychiatry and protein chemistry. And it's unlikely or probably impossible for any single person, no matter how brilliant and skilled they are, to obtain answers to these difficult problems. To do that, we need to all work together.
What would be your recommendations for your patients then?
I have a series of recommendations which are not proven by any double-blind, placebo-controlled, randomized trial. However, nothing that I recommend could possibly be bad for anyone. And these are for my patients who do have Alzheimer's disease or mild cognitive impairment or Parkinson's disease, as well as actually healthy people. That is, people should have a high-fiber diet with a diverse amount of fruit and vegetables, and generally not meat. And they should be physically active, mentally, socially, emotionally and spiritually active throughout life. And these things together with a diverse diet composed of diverse good things. For example, brown rice has 5 times more fiber than white rice. And we all believe that a high fiber diet leads to relatively less inflammation in the body, and inflammation is an important component of Alzheimer's, Parkinson's, and the other conditions, as well as stroke and cancer.